I'm going to tell you about symbiosis story, which is a biological concept, and try to convince you that actually it has turned into a computer science concept. So what is symbiosis? Symbiosis is a biological association which is lasting and mutually beneficial, even essential for their survival between two organisms. So when two people are in symbiosis, as you can see there, they behave as if they were one complete person. And we can think about a few couples, as you see, but usually scientists refer to symbiosis when they describe interactions and associations between two living species in the world. So the main example that you may have heard about symbiosis has been popularized in the cartoon Finding Nemo. Maybe you've heard about that, where you saw maybe that these clownfish lives in groups around their home, which is an anemone, and they are insensitive to the poison in the particle of the adenoma, so that they can hide in it and hide from predators. And in return, what happens is that they provide the anemone with their waste, and the anemone then grows faster than the other ones. So symbiosis is very common. We are made of symbiosis because it has structured the evolution and it appears everywhere, but it's quite difficult to elucidate and to understand. An example of symbiosis has been applied here in Chile, for instance, in the industry. You may know about the bioleaching process, which consists in extracting ore from mines and then just immerse it into a soup of bacteria. And those 5 bacteria has been chosen carefully by biotechnologists because they have realized that they were all efficient, more efficient together than alone. So finally, you just wait for the ore to decompose itself, and it provides copper. What is interesting here is that actually it took us 3 years in a collaboration with the Center of Mathematical Modeling here in Chile to try to figure out why those 5 bacteria were so efficient together. And the main hypothesis that we have now is that the orange bacteria provides the other one with NADP, something that you can buy. It's a cofactor that you can buy in drugstores, actually. And the others, and it provides the other bacteria with NADP, and conversely, the other bacteria help the orange bacteria to grow. So finally, what happens there is that symbiosis is everywhere, but it also is in your body. And this is the main point that has been discovered in the last 15 years. We are living thanks to the symbiosis with all the microbes, which we call the microbiota, placed in our intestinal stuff. And also plants on the left side, on the right side, live thanks to the microbes which live around the root of the plants. So it means that we are not sure that microbes are completely inherent to our life. And naturally, scientists want to use this property in order to have more impact on health in particular. For instance, it has been shown that if you look at intestinal diseases such as the Crohn's disease, manipulating the microbiota, your intestinal flora microbiota, can be a key target for medicine. We already manipulate microbiomes by eating probiotics, for sure, but we could do better, for sure, and this is what scientists are working on. But the point is, at the moment, is that we know that microbiota are important, but we don't know how much they are and especially why they are so important. And to do that, we need to come back and take a step forward from humans and use the capability of investigating microbiomes of plants in order to make new experiments and gain new understanding. This is quite natural in biology because you may have known the general scheme of discovery in biology. First, what we do in biology is undergoing a naturalist approach, which consists in observing everything that you have around and inside. This follows Darwin's teaching. And then you make hypotheses, and as soon as these hypotheses deduced from the foundation are contradictory with what we know, with dogmas, then you want to make new discoveries and prove them. And this is where biologists follow Pasteur's approach, which is an experimental approach, in order to prove hypotheses. You can imagine that we cannot do any hypothesis on humans. It would be a little bit non-ethical. So that people now are searching and making microbiota hypotheses on non-human species. And an example of experiment is an experiment which has been done 10 years ago in Station Biologique de Roscoff in Western France. It's a Balearic city in Western France. And they considered their most favorite algae on the left, which is Ectocarpus siliculosus, and they just immerged it into a bath of antibiotics. And what you see on the right is that the algae doesn't die. That's the first observation, but it has clear problems to grow. I mean, this algae really cannot grow easily. So it's a proof that the microbes which were killed by antibiotics are important for algal growth. Okay? But if we want to move forward this discovery in biology process, then we need to make experiments in order to figure out what are those bacteria which were killed and which were important for the algae. And so here we are puzzled, because you can imagine that you have hundreds of bacteria which are living in symbiosis with the algae. And if you want to pick up the few ones which may have an impact, then you would have to test each bacteria one by one, added to the algae, and see whether it has an impact. And if there are combinations of bacteria which are useful, then you should have to test all combinations of bacteria. This makes millions of experimentations, and this is not possible. So either we stop our study, or we take a different perspective, which is what was done, because actually what we know about these bacteria is that now we have information about their genomes. And what I want to convince you is that playing with genomes, they are making computer science and artificial intelligence, is a huge playing field to have more insight on biology. So if you take this bacteria, you have this genome, and as soon as you have a genome, it's a DNA sequence, you can use computer science, some softwares, bioinformatics, some softwares, which allow you to figure out which are the parts of the genome, so the genes, which encode some macromolecular, 3D macromolecules, which are called proteins. And among them, what we like are those proteins, which are called enzymes, and they have a very specific shape, because they are huge for the living world. And inside, they have a small part, which is just like a key, and a key specific to small molecules, which are called metabolites. Those metabolites has everything that you eat daily. So what happens in a cell is that as soon as an enzyme is present, and the metabolite is around it, the enzyme grabs the metabolite and just torture it, clearly. And as it has tortured it, it's going to release a new metabolite slightly transformed. It can have a new atom or something, or it can have another atom. And this slightly transformed metabolite also can be grabbed by another enzyme, which is going to release a new metabolite, and so on and so on. So what you get at the end is a set of transformations, the most famous of which is the Krebs cycle, okay? But actually, we don't have not only 10 enzymes in your body, what we have is a map of transformation. So the Krebs cycle is just in the middle there. And this is a description at the macromolecular level of everything that could happen in a cell, so, and that you may have learned in terms of cellular respiration. The important thing is that as soon as you have a genome, you may build such a map, okay? And as a computer scientist, then we are done, because we have a map, and as soon as we have a map, we can play with it. So for instance, if you want to model and elucidate, figure out how bacteria grow, you just have to consider every entries which are these metabolites which are in the culture medium, and look at paths in the map, which are going to provide new compounds, allowing the bacteria to grow, and even side products such as ethanol, vitamins, or insulin. So we are done now. If we want to model the growth of a bacteria, we just have to find the path. But as you have many ways to go from South Santiago to North Santiago, you can imagine that there are many ways to cross all this map. And then this is the magic for me of metabolism, is that we have a metabolism entropy principle available, which means that among all these paths which are possible, evolution has made such that the bacteria will choose the most efficient path, allowing the bacteria to grow. Not even, so just going to select a single one, and this path is something that we can identify thanks to mathematical equation. So we have turned a growing issue, biological issue, into solving mathematical inequations, and this is very, very nice for me as a mathematician or a computer scientist. And this is why we usually say that discovery in biology has switched from this experimental approach from Pasteur to a modern approach, which is a data science approach based on DNA discovery made by Rosalind Franklin. It's quite funny because when you speak with life scientists, they just consider data science as a nightmare, nothing else. And when you speak to computer scientists, they consider data science in biology as a paradise, because these data are absolutely awful. I mean, they are much more difficult to study than social media data or astronomical data, because they are full of errors, of bias, of uncertainty, and this generates huge challenges for computer scientists. If I come back to my question of bacteria, remember, I wanted to select those bacteria that I could test as an experiment. Then it can be transcribed into a new problem, which is a map problem. You have the map of the algae on the left. You have the many maps of the bacteria, which hundreds of maps. Each is different. It has been reconstructed from their genomes. And at the end, what you want is just to pick up the smallest number of maps in the middle, which allows to complete and to extend the algae map, so that biomass will be improved mathematically. So this is a new problem. It's a new computer science problem. It's not that easy to solve. And what we did with several colleagues was to use what we call old-fashioned artificial intelligence, which is something allowing to mimic reasoning process. So the small program that you can see here is nothing else than encoding rules as if-then-else rules. So maybe you can see it. I don't have a pointer, but in the second line, you can see that you read from the right to the left, and if you see that if the crime did hold in the kitchen, then it should have been with a candlestick. But you also know that if the murderer was a Colonel Mustard and the weapon was not a revolver, then it should have held in the hole. Okay? So this is just a Cluedo game, and you can imagine that the program that we are playing with just takes all these constraints into account and solves and provides you with the answer according to what you know. We do exactly the same with this algae. We just say that if a bacteria has an enzyme and sees some metabolites, then the released metabolites should be considered as predictable, and so on, and so on, and so on. And then we look for the best solution among everything which is predictable. So it's not that easy, actually. It makes a huge computer science program, but it works. And for instance, if we come back to the algae, we predicted 3 bacteria that could be added to the algae in order to restore the biomass. And you can see on the right-hand side the result of the experiment. Actually, the algae grew, which was something for me as a miracle. it was not possible that at the first step of an output of an algorithm, the output was true. It was true, but nearly true, because biologists, just to be safe, looked at all these bacteria which were around the grown-up algae, and they found a new bacteria that should have not been there. So it means that finally there may have been a bacteria which was resistant to antibiotics, and he has hidden itself under the wall in the bath of antibiotics, and just get benefit of the new bacteria that we added to grow again and kill the other one. So finally, the algae is not now to be in symbiosis with its microbiome, but what is clear now is that what's taking place inside microbiome is just something related to competitions and kill and death process, which is not symbiosis at all, actually. There are many mathematical models to undergo on this question. So what's next? We have to move forward again and again, move at larger scales again and again, since now we are going to have macrobiomes, which means hundreds of maps for each patient of each illness or each plant in any agriculture experiment. And what we want to do is to compare all this set of maps in order to figure out what are the smart differences between them which can explain healthy or disease. So this is where deep learning is starting to be a wonderful playing field. We are currently working on that, but what the main issue is that it will not provide us with explanations. We will be able to discriminate, but we will not be able to understand why things are going on. So it's clear that there are still more computational issues and AI problems to solve before moving forward, and I hope that maybe in 10 years I will come back here to explain you the follow-up of this talk. Thank you.